So our next talk will be by Matt Stone, and then he's going to talk about benchmarking network inference methods from single-cell RNA-seq data.
Thank you very much. Hi, I'm Matt Stone from the University of Wisconsin, and today I'm going to be sharing our work to evaluate some current methods to infer regulatory networks from single-cell expression data. Gene expression is regulated in large part by transcription factors whose physical binding to gene promoters can activate or repress transcription. The relationships between transcription factors and their targets form a genome-wide network of regulatory interactions which govern important cellular processes such as differentiation or immune response. An ongoing challenge in regulatory genomics is the inference of the structure of these regulatory networks directly from cheaply available expression data. Historically, expression data was measured in bulk using microarray or RNA-seq, but this presented a sample size problem as an experiment would yield only a handful of expression measurements per gene, and researchers had to collect and aggregate enough data sets to infer regulatory network structure. With the advent of single-cell RNA sequencing, in addition to all the applications we've seen towards problems like dissecting cellular heterogeneity or development, we have also effectively solved this sample size problem, as we're now routinely generating data sets with thousands of genes with a single experiment. And so we now have the big expression data that we need to infer regulatory networks. And as a consequence, we've seen a rapid proliferation in methods to infer regulatory networks in the single-cell context. And so we wanted to evaluate how they're all performing. So we sought to address a few open questions in network inference. First, we wanted to know how these algorithms scale to these large data sets that we're seeing with thousands of cells. We wanted to assess how accurate the inferred networks are as compared to orthogonal sources of evidence, such as chip or gene knockdown experiments. We wanted to know whether the networks these methods are inferring are similar, if they're predicting similar interactions. And we wanted to know if imputing sparse single-cell expression data can improve the accuracy of inferred networks. So we evaluated a dozen algorithms to infer regulatory networks from a variety of inference paradigms, including graphical models, information theoretic methods, differential equations, correlation. And in addition to 11 published algorithms, we also constructed a network using experiment-wide Pearson's correlation to weight interactions. We used 6 publicly available data sets to evaluate the algorithms. In addition to a smaller yeast data set, we included 5 mammalian data sets that we thought were well representative of current single-cell experiments, ranging from a few thousand cells up to 36,000 cells. And all the data sets had roughly the same proportion of the genome sufficiently expressed. So as a brief overview of our pipeline, we first downloaded all the expression matrices from Geo and performed some basic processing, such as cleaning of gene names. Prior to running the algorithms, we depth normalized and square root transformed the expression counts. Some algorithms required cellular trajectories, which we learned using Monocle. And finally, we imputed the value of the missing expression counts in each matrix using magic. And then we applied each algorithm to both the normalized and the imputed data sets and compared them against a set of species-specific gold standards. So first, we looked at how these algorithms scale up. We took a data set of about 5,000 human embryonic stem cells and generated subsets of the genes in the data set from 10 to 8,000 genes, and measured the runtime required by each algorithm as you scaled up the size of the potential network and the number of interactions you have to test. We found that to infer a full genome-wide network, about a third of the algorithms could do so in under a day, about a third in under a week, and a third took a bit longer than that. And as a consequence of some of these runtime constraints, we excluded BTR and SHRM from the remaining analyses. Similarly, we measured memory consumption as you grew the size of your potential network. And while most algorithms were able to run using less than 16 gigabytes of memory, which is pretty reasonable, we had to exclude hurdle normal because due to the memory we projected it would require for a full genome-wide network. So after inferring networks with all these algorithms, we sought to evaluate how accurate they are. And there's a number of orthogonal sources of experimental evidence for regulatory interactions, such as chip experiments, which identify transcription factors that are physically binding to a promoter region, or perturbations of transcription factors, such as knockdown or overexpression experiments. And we curated 4 gold standards for each species using a mix of chip and transcription factor perturbation methods. For the mammalian datasets, we also curated a gold standard of interactions reported in the literature. And I did want to briefly highlight the differences in size of these gold standards. The literature network in particular is quite sparse, so some of the accuracy measures that we'll see on the coming slides won't be directly comparable between gold standards. So as most of you are aware, a common method to evaluate network accuracy is to use precision recall against the gold standard. If you start with your top set of edges and gradually increase the size of your network, you'll monotonically increase recall, and you can plot how precision changes as you incorporate new edges. And the area under the PR curve is very common for evaluating imbalanced classification problems such as this. So there are a lot of bars on this plot. They're all representing the height of each is representing the AUPR of an inferred network with respect to a given gold standard. In the top left, we have yeast, and you can see that here, Scode is doing well on the knockdown gold standard in yeast, but kind of everywhere else, there's not a lot of distinction between how the algorithms are performing relative to one another. But AUPR is measuring the accuracy of the entire network, and frequently a researcher is not concerned with the global network, but a set of top interactions. And so then we might want to focus on the precision recall of a specific subset of high confidence edges. And when you look at how these algorithms are performing as you increase the number of top edges, as you expect, recall goes up across the board, but with precision, you see a much clearer stratification between the algorithms, particularly when you consider the top 50,000 or fewer edges. And so we computed the F-score on the top 50,000 edges for each algorithm, and we saw a much clearer delineation between the algorithms. So Scode remains the top performer on the yeast dataset. And we saw it also did well against one of the chip gold standards in 2 of the mammalian datasets. But across the board for the mammalian datasets, especially with regards to the interactions predicted by knockdown, we see that PIDC and the network constructed using Pearson's correlation have the highest accuracy. And we see that these 2 algorithms are also top performers for chip-based gold standards in the mammals, as well as, you might have to squint a little bit, but towards the literature interactions as well. But interestingly, we see that SIL-GGM, despite not performing as well on some of the other gold standards, is fairly consistently superior to the other methods with respect to the gain-of-function interactions. So AUPR and the F-score are helpful metrics to get a single measurement for how accurate an entire network is. But sometimes we might want an additional level of granularity, and we might want to ask how well a network is predicting the activity of a specific transcription factor. And so if there's 5 possible targets in the genome for a transcription factor, and the gold standard includes edges to a few, your inferred network predicts edges to some of those and some to others. It might overlap well, it might overlap worse. But you can quantify how well your algorithm is inferring the activity of this transcription factor by computing a sort of enrichment ratio, saying the fraction of true edges that you're inferring as contrasted to random. And you can assign a p-value using a hypergeometric test. So on the next slide, we'll see on the left, we have a heat map of all the enrichment ratios of several transcription factors. This is looking at one of the mouse embryonic stem cell datasets with respect to the chip-based gold standard. In the interest of time, I won't be going exhaustively through all of them. But you can see that most of the algorithms are identifying transcription factors that are known to be implicated in pluripotency. On the right, we can take the number of transcription factors with significant overlap after FDR correction and count them and use that as yet another measure to compare the algorithms. And using this here, you can see that SylGGM is again performing well on the gain-of-function relationships, and we see some evidence that Merlin, a bulk algorithm, is doing well on a chip-based gold standard. So we also wanted to know how similar the networks inferred by the different methods were. Here I'm showing a heat map of Jacquard similarity between each inferred network in the top 50,000 edges of each network in each data set. And you can see that the strongest overlap is shared by PIDC and Pearson in some of the mammalian data sets, which is consistent with their strong performance there. But you can also see that the correlation-based methods, PIDC, Scenic, Merlin, are sort of forming a family that are generally inferring similar interactions. And you can also notice that Kane and Dremy is inferring a distinctly different set of interactions. So finally, we want to determine whether imputation could improve network accuracy. Single-cell data is characterized by a large number of zero-valued expression counts, a technical artifact known as dropout. Several methods exist to try to impute the values of these missing counts. We applied an algorithm called MAGIC, which very simply computes pairwise similarity between cells and attempts to redistribute expression counts among similar cells. And when you apply it, you can see that the expression patterns for each gene are sort of like smooth consistently across the cells in the data set. So we evaluated the AUPR of the inferred network using both the normalized data, as we saw previously, and using the imputed data. Here, to simplify things, each data point is one of the inferred networks with respect to a gold standard that we were seeing in the previous slides. Anything below the y equals x line was not improved by imputation, and anything above it benefited from it. In the Pearson correlation network, we didn't see much improvement. And when we zoomed out and considered all the algorithms, we also didn't see much difference when using AUPR as our metric. When we instead considered the top 50,000 edges, we saw a little more of an extreme effect. In general, imputation is not improving the accuracy of the top 50,000 or so edges in these networks, with the noticeable exception of S-code. But the remaining algorithms are not clearly benefiting from it. So to conclude, AUPR doesn't distinguish a clear best inference method, but when you instead consider a top set of interactions, you can identify algorithms which are more or less accurate towards our gold standards, although that varies based on which gold standard you're comparing to. In the mammalian data sets, we saw that correlation in PIDC. We're demonstrating the highest accuracy towards CHIP and our knockdown-based gold standards. We found that imputing sparse expression counts generally doesn't improve the accuracy of the inferred networks. And we think this demonstrates a need for some improved gold standards that are specific to single-cell data. And with that, I'd like to thank Sushmita and her team, who've been very kind and helpful throughout all of this and guiding this project. I'd like to thank NLM and RegSys for whose generosity allowed me to be here today, my wife Amanda, through whom all things are possible, and all the researchers that have poured time and money into sequencing and processing these data sets and then making them readily available for other people to reuse, and the developers who have put their methods out here to be used and scrutinized by us. Honestly, every method that we looked at, installation was very straightforward. They were all well documented, which I think is a testament to the reproducibility in this community. So, thank you.
Questions?
Yeah, just one quick question. Have you tried ensembling, pooling the predictions and see how much they improve?
We did do that. I haven't done it exhaustively. We've tried a couple methods. One where you take like the top 50,000 edges, average their edge weights. We've tried applying like the border voting method to generating an ensemble, and we haven't found that it is outperforming any of the individual algorithms. Yeah.
You assembled all of them?
Yeah. So we took all 11.
That might not work, but the 3 that are highly correlated, I mean, relatively CV.
We have not tried that. That's a very good suggestion.
Hi, interesting talk.
Thank you.
In your Jacob score comparison plot for all algorithms, so it hardly seems there is a score similarity of 0.6, if I get it correct. So did you explore any of the method in more detail, that which edges are more meaningful in biological aspects?
We have not delved into that yet. We are interested in doing that. We want to strike kind of a balance between evaluating the performance of all these algorithms without going too far into reanalyzing all the data sets. But we're definitely interested in, like, especially now that we found some algorithms are relatively similar, trying to investigate some of the top edges.
Yeah. Thank you.
Yeah.
Thanks for the talk. So all of your comparisons are based on what you call your gold standards, right?
Yes.
But how confident are you in these gold standards?
That's
I mean, if you look at ChIP-seq, I mean, it's not because you have a peak in the promoter that it will be regulating this gene. If you look at knockdown, you will have a lot of indirect targets, right? So how much do you think this would influence your results?
Entirely. I totally agree, and that's why I'm very, I don't like to use the word like best when trying to describe like the performance of these algorithms, because it is so depending on the assumptions that you're making, the gold standards that you're using. Like we saw here that if that like the performance of an algorithm will vary wildly depending on what you're looking at it, and we didn't look at just these 4. We looked at different like subsets of them, and we saw like similar variability across. So it's definitely susceptible to that.
One question here, one question here.
Yeah, quick question. PIDC and correlation doesn't give us directionality. So how did you compare with other algorithms?
All of these are using unsigned edges.
You ignore the directionality.
Yeah, well, it's not ignoring the direction. We restricted all the networks to a set of known transcription factors and considered all the edges where a known transcription factor was a regulator and some other gene was a possible target. And then we only considered the magnitude of the edge weight. So we're just hoping to find strength of signal of an interaction rather than looking at whether a transcription factor might be upregulating or downregulating a gene.
Thank you.
Very nice talk. I was wondering whether you plan to include also generative models like autoencoders, as it seems that the last layer of the decoder, for instance, a variation autoencoder is able to retrieve regulatory modules of the association between genes.
I don't think we've looked at any specifically yet, but we're definitely open to considering like further algorithms to include in this comparison.
All right, so let's thank the speaker one more time.